Introduction to Evolutionary Thought. The concept of evolution, the idea that species change over time, has fundamentally transformed our understanding of the natural world. Before the modern framework of evolutionary theory emerged, many cultures had their own explanations for the diversity of life. Early ideas about species change laid the groundwork for what would become the theory of evolution, but it was not until the 19th century that a coherent, scientifically accepted theory began to take shape. Historically, many early thinkers speculated about the possibility of species transformation. In ancient Greece, philosophers like Anaximander proposed that life evolved from simpler forms. Similarly, the philosopher Empedocles suggested that life arose from a combination of different elements. However, these early notions were not based on empirical evidence and lacked a systematic approach. During the Renaissance and Enlightenment periods, as scientific inquiry advanced, so did ideas about species change. One notable figure was Georges-Louis Leclerc, Comte de Buffon, who proposed that species could change over time, although he did not fully articulate a mechanism for this change. Buffon's ideas influenced later thinkers, but it was Jean-Baptiste Lamarck who took a significant step toward a more formal theory of evolution. Lamarck proposed that species evolved through the inheritance of acquired characteristics, a concept suggesting that traits acquired during an organism's lifetime could be passed on to offspring. Despite Lamarck's innovative ideas, his theory was ultimately challenged. The notion of evolution itself, though increasingly considered, was still met with skepticism, largely due to the lack of a concrete mechanism explaining how evolutionary change occurred. It was within this context of inquiry and debate that Charles Darwin emerged as a pivotal figure in the history of evolutionary thought. Charles Darwin, born in 1809, was deeply influenced by his voyage on HMS Beagle, which began in 1831. The journey allowed him to observe a variety of species and their adaptations to different environments, particularly in the Galapagos Islands. During his voyage, Darwin encountered numerous examples of species exhibiting variations adapted to their specific environments. These observations sparked his thinking about how species might change over time. Darwin's exposure to the work of other scientists, such as Thomas Malthus, who discussed population growth and resource limitations, further shaped his ideas. Malthus's essay on the principle of population highlighted the struggle for existence and competition for resources, which Darwin saw as central to the process of evolutionary change. Upon returning to England, Darwin meticulously analyzed his observations and began formulating his theory of natural selection. However, he was not alone in his revolutionary thinking. Alfred Russel Wallace, another naturalist, independently arrived at similar conclusions regarding evolution and natural selection. Wallace's correspondence with Darwin, presenting his own ideas, prompted Darwin to publish his findings. In 1859, Darwin's seminal work, On the Origin of Species, was published. This book presented a detailed argument for natural selection as the mechanism of evolution. Darwin proposed that species evolve over time through a process where individuals with advantageous traits are more likely to survive and reproduce, passing those traits to future generations. This concept of natural selection provided a robust explanation for the diversity of life observed in nature. Darwin's theory was groundbreaking but also controversial. It challenged the prevailing view of the time, which often held that species were immutable and created independently. The acceptance of Darwin's theory was gradual and met with resistance from various quarters, including religious and scientific communities. However, over time, the accumulation of evidence from multiple scientific disciplines, such as paleontology, comparative anatomy, and genetics, solidified the theory of evolution as a cornerstone of modern biology. In conclusion, the historical context of evolutionary thought reveals a fascinating journey from early speculations to the establishment of a comprehensive theory. Darwin's work, building upon and refining earlier ideas, marked a pivotal moment in our understanding of the natural world. The theory of evolution not only revolutionized biology, but also set the stage for ongoing research into the mechanisms of natural selection and the dynamic processes that shape life on Earth. Charles Darwin and the foundations of evolution. Charles Darwin stands as a central figure in the history of evolutionary biology, credited with formulating and popularizing the theory of natural selection. His groundbreaking work fundamentally altered our understanding of life's complexity and diversity. To grasp the significance of Darwin's contributions, it's essential to explore his early life, the pivotal voyage of HMS Beagle, and the subsequent development of his revolutionary theory. Born on February 12, 1809, in Shrewsbury, England, Charles Darwin came from a well-to-do family with a strong scientific inclination. His father, Robert Darwin, was a physician, and his grandfather, Erasmus Darwin, was a noted poet and naturalist who had earlier speculated about evolutionary ideas. Initially, Darwin was enrolled at the University of Edinburgh to study medicine but found himself disinterested in the subject. His true passion lay in natural history, and he eventually pursued studies in this field at Cambridge University, where he was mentored by the geologist Adam Sedgwick and the botanist John Stevens Henslow. In 1831, Darwin's life took a decisive turn when he was offered a position as a naturalist on HMS Beagle, a British naval vessel embarking on a voyage to survey the coasts of South America. This voyage, which lasted nearly 5 years, became the crucible in which Darwin's ideas about evolution were forged. The Beagle's journey allowed Darwin to explore diverse ecosystems and collect a wealth of specimens, including plants, animals, and fossils. One of the most significant aspects of the voyage was Darwin's observations in the Galapagos Islands, a remote archipelago in the Pacific Ocean. Here, Darwin noted the variations among species on different islands, particularly the differences in the beak shapes of finches that seemed adapted to different food sources. These observations were pivotal, suggesting to Darwin that species were not fixed but could adapt to their environments over time. Darwin's voyage also exposed him to the broader patterns of biogeography and geology. The evidence he collected, such as the similarity of fossils from South America to those of living species in the same region, supported the idea that species were related and had evolved from common ancestors. Additionally, observations of geological formations and fossil layers helped him understand the gradual changes in Earth's history, reinforcing the concept of slow and gradual processes. Upon returning to England in 1836, Darwin embarked on an extensive period of analysis and reflection. He meticulously reviewed his findings, consulted with fellow scientists, and began to piece together the mechanisms behind the observed variations in species. Darwin was deeply influenced by Thomas Malthus's essay on population growth, which argued that populations tend to grow faster than the resources available to sustain them. Malthus's idea of the struggle for existence provided Darwin with a crucial insight into how competition for resources could drive evolutionary change. In 1844, Darwin wrote a private essay outlining his theory of natural selection, but he delayed publication, uncertain about how his ideas would be received. The situation changed dramatically in 1858 when Alfred Russel Wallace, a fellow naturalist working in the Malay archipelago, independently arrived at a similar theory of natural selection. Wallace's correspondence with Darwin spurred him to act. In 1859, Darwin published On the Origin of Species by Means of Natural Selection, a comprehensive account of his theory. On the Origin of Species presented the idea that species evolve over time through the process of natural selection, where individuals with advantageous traits are more likely to survive and reproduce. This mechanism explains how species adapt to their environments and how new species arise. Darwin's arguments were supported by a plethora of evidence, including observations of variation within species, the fossil record, and biogeographical patterns. Darwin's theory faced considerable resistance from some quarters, particularly from those who adhered to creationist views. However, over time, the accumulation of additional evidence from various scientific disciplines, such as genetics and paleontology, has reinforced and expanded upon Darwin's original ideas. In summary, Charles Darwin's contributions to evolutionary biology were monumental. His voyage on the Beagle, combined with his meticulous analysis and the development of his theory of natural selection, established a new framework for understanding life's diversity. Darwin's work laid the foundation for modern evolutionary biology and continues to influence our understanding of the natural world. Principles of natural selection. The theory of natural selection, articulated by Charles Darwin, represents a cornerstone of evolutionary biology. It offers a compelling explanation for how species adapt to their environments and how evolutionary change occurs over time. This section explores the core principles of natural selection, which are variation, heritability, the struggle for existence, and differential reproduction. Variation. Variation is the first principle of natural selection and refers to the differences in traits observed among individuals within a population. These traits can include physical characteristics such as size, color, and shape, as well as behavioral traits. Variation arises due to genetic differences, which are a result of mutations, genetic recombination during reproduction, and other genetic processes. In any given population, individuals exhibit a range of traits. For example, in a population of beetles, some might be green while others are brown. This variation is crucial because it provides the raw material for natural selection. Without variation, there would be no differences for natural selection to act upon, and adaptation would not occur. Heritability. Heritability is the principle that traits passed from parents to offspring are influenced by genetic factors. For natural selection to drive evolutionary change, the traits that confer advantages must be heritable. In other words, traits beneficial for survival and reproduction must be able to be transmitted from one generation to the next. Heritability is determined by the genetic makeup of individuals. When an advantageous trait is present, it is encoded in the DNA and thus can be inherited by offspring. For example, if a certain coloration provides camouflage that helps an animal avoid predators, this trait can be passed down through generations if it is genetically controlled. The ability of these advantageous traits to persist and spread through a population is fundamental to the process of natural selection. Struggle for existence. The struggle for existence is a principle that highlights the competition among individuals within a population for limited resources such as food, shelter, and mates. Resources are finite, and not all individuals can survive and reproduce. This competition creates a selective pressure on the traits of individuals within the population. Darwin borrowed the concept of the struggle for existence from Thomas Malthus's essay on population dynamics. Malthus argued that populations grow exponentially while resources grow arithmetically, leading to competition and scarcity. In nature, this struggle manifests in various ways, such as competition for food, territory, or mates. Those individuals who are better adapted to their environment have a higher chance of surviving and reproducing, while others may not. Differential reproduction. Differential reproduction is the principle that individuals with advantageous traits are more likely to survive and reproduce, thereby passing these beneficial traits to their offspring. This principle is at the heart of natural selection, as it explains how certain traits become more common in a population over time. In any given environment, some traits confer a survival advantage. For example, in a predator-rich environment, faster prey animals might have a higher chance of escaping and thus reproducing more successfully. Over time, the traits that enhance survival and reproductive success become more prevalent in the population. This differential reproductive success leads to the gradual evolution of the population as advantageous traits become more common. Conclusion: the principles of natural selection, variation, heritability, the struggle for existence, and differential reproduction provide a framework for understanding how species evolve and adapt over time. Variation within populations provides the diversity needed for selection. Heritability ensures that advantageous traits can be passed on to future generations. The struggle for existence creates the selective pressures that favor certain traits. Finally, differential reproduction leads to the accumulation of beneficial traits in a population. These principles together explain how natural selection drives the process of evolution, shaping the diversity of life on Earth. They offer a scientific basis for understanding how species adapt to their environments and how evolutionary change occurs over generations. The theory of natural selection not only deepens our comprehension of biological processes, but also highlights the dynamic interplay between organisms and their environments. The fossil record as evidence for evolution. The fossil record is a critical source of evidence supporting the theory of evolution, providing a historical record of life on Earth and documenting the gradual changes in species over time. Fossils, the preserved remains or traces of organisms from the past, offer insights into the evolutionary process, including the emergence of new species, the adaptation of organisms to different environments, and the extinction of species. This section explores how the fossil record supports evolutionary theory and highlights key examples that illustrate its significance. Understanding the fossil record. The fossil record consists of remains or traces of ancient organisms preserved in sedimentary rock layers. Fossils can include bones, shells, teeth, and even impressions of plants or animals. The process of fossilization typically requires rapid burial and the accumulation of sediment, which protects the remains from decay and physical disruption. Over millions of years, the sediments become rock, and the organic materials are replaced by minerals, creating a fossil. Fossils are found in sedimentary rock layers, which are arranged chronologically. Lower layers represent older geological periods, while higher layers are more recent. By studying the distribution of fossils in these layers, scientists can reconstruct the history of life and identify patterns of evolutionary change. Transitional fossils. One of the most compelling aspects of the fossil record is the presence of transitional fossils, fossils that exhibit traits intermediate between two different groups of organisms. These fossils provide direct evidence of evolutionary change and the gradual transformation of species over time. A classic example of a transitional fossil is Archaeopteryx, a late Jurassic species that displays characteristics of both reptiles and birds. Archaeopteryx had feathers and wings like modern birds, but also had teeth, a bony tail, and clawed fingers, features typical of reptiles. This combination of traits supports the hypothesis that birds evolved from theropod dinosaurs. Another example is Tiktaalik, a transitional fossil between fish and amphibians. Discovered in 2004, Tiktaalik had features of both aquatic and terrestrial vertebrates. It possessed fins with wrist-like structures, suggesting an evolutionary adaptation to life on land. Its discovery filled a significant gap in our understanding of the transition from water to land. Gradual changes and speciation. The fossil record also demonstrates gradual evolutionary changes within lineages, providing evidence of how species evolve over time. For instance, the evolution of horses illustrates this gradual change. Fossils of early horse ancestors, such as Hyracotherium, formerly known as Eohippus, show small, multi-toed animals with low-crowned teeth adapted for browsing on soft vegetation. Over time, these ancestors evolved into larger, single-toed horses with high-crowned teeth suited for grazing on grasses. The fossil record documents these changes, illustrating the process of gradual adaptation to changing environments. Patterns of extinction and radiations. The fossil record also reveals patterns of extinction and the emergence of new species. Mass extinctions, such as the one that ended the reign of the dinosaurs approximately 66 million years ago, had profound effects on the course of evolution. The extinction of the non-avian dinosaurs paved the way for the diversification of mammals and the eventual rise of human ancestors. Similarly, the phenomenon of adaptive radiation is well documented in the fossil record. After major extinctions or the colonization of new habitats, species often undergo rapid diversification to fill available ecological niches. The evolution of the Darwin's finches on the Galapagos Islands is a modern example of adaptive radiation, where different species of finches evolved from a common ancestor to exploit various food sources. Conclusion. The fossil record provides a rich and detailed account of the history of life on Earth, offering compelling evidence for the theory of evolution. Transitional fossils reveal intermediate forms between major groups, illustrating the gradual nature of evolutionary change. The documentation of gradual changes within lineages and patterns of extinction and adaptive radiation further supports the idea of evolution as a dynamic and ongoing process. Through the study of fossils, scientists continue to uncover the intricate history of life and deepen our understanding of the mechanisms driving evolutionary change. Comparative anatomy and embryology. Comparative anatomy and embryology are two critical fields that provide substantial evidence for the theory of evolution. By examining the anatomical structures and developmental processes of different organisms, scientists can uncover patterns that suggest common ancestry and evolutionary relationships. This section explores how comparative anatomy and embryology support the concept of evolution and highlight the interconnectedness of life. Comparative anatomy. Comparative anatomy involves comparing the anatomical structures of different species to identify similarities and differences. One of the key concepts in comparative anatomy is homology, which refers to structures that are similar in different species due to shared ancestry. Homologous structures provide evidence of common descent and evolutionary relationships. A classic example of homologous structures is the limb bones of vertebrates. The forelimbs of humans, cats, whales, and bats, although adapted for different functions such as grasping, walking, swimming, and flying, share a common underlying bone structure. These similarities in bone arrangement, humerus, radius, ulna, carpals, metacarpals, and phalanges suggest that these diverse species evolved from a common ancestor with a similar limb structure. In contrast, analogous structures are similar in function but do not share a common evolutionary origin. For example, the wings of insects and birds are both adapted for flight but evolved independently. The similarity in function but not in underlying structure highlights the difference between homology and analogy, reinforcing the idea that homologous structures are indicative of shared ancestry. Another significant concept in comparative anatomy is vestigial structures, which are remnants of organs or structures that had important functions in ancestral species but are now reduced or non-functional. Examples include the human appendix, which is a vestigial remnant of a larger cecum used for digesting cellulose in herbivorous ancestors, and the pelvic bones in whales and snakes, which are remnants of hind limbs. The presence of vestigial structures supports the idea of evolutionary change and adaptation over time. Embryology. Embryology, the study of embryonic development, provides additional evidence for evolution through the examination of developmental similarities among different species. During early stages of development, embryos of different species often exhibit remarkable similarities, reflecting their common ancestry. For instance, vertebrate embryos, including those of fish, amphibians, reptiles, birds, and mammals, all possess pharyngeal pouches and tail structures at certain developmental stages. These similarities are a result of shared ancestry and indicate that these species diverged from a common progenitor. The presence of gill slits in the embryos of all vertebrates, which are later modified or lost in most species, provides insight into the evolutionary transitions from aquatic to terrestrial environments. Comparative embryology also reveals patterns of developmental divergence and convergence. As embryos develop, species may exhibit similar early developmental stages, but diverge in later stages, leading to the distinct forms observed in adults. For example, the early embryonic development of all vertebrates shows a similar body plan, but as development progresses, the embryos differentiate into species-specific forms. This process of developmental divergence reflects evolutionary changes and adaptations. Developmental homology further supports evolutionary theory by showing how similar developmental processes can lead to diverse adult forms. The molecular and genetic mechanisms governing development are conserved across species, suggesting that fundamental developmental pathways were inherited from a common ancestor and modified over time to produce various forms. Conclusion: comparative anatomy and embryology provide compelling evidence for evolution by revealing patterns of similarity and divergence among different species. Homologous structures and vestigial organs demonstrate shared ancestry and evolutionary change, while similarities in embryonic development reflect common origins and developmental pathways. These fields underscore the interconnectedness of life and the gradual process of evolution that has shaped the diversity of organisms we observe today. Through comparative anatomy and embryology, scientists gain valuable insights into the evolutionary history of species and the mechanisms driving their development and adaptation. Molecular biology and genetic evidence. Molecular biology and genetics have revolutionized our understanding of evolution, providing detailed insights into the underlying mechanisms driving evolutionary change. By examining the molecular and genetic similarities among organisms, scientists can uncover evidence of common ancestry, evolutionary relationships, and the processes that shape the diversity of life. This section explores how molecular biology and genetic evidence support evolutionary theory and contribute to our understanding of the evolutionary process. Molecular homologies. Molecular homologies refer to similarities in the molecular structures of different organisms that arise from shared ancestry. One of the most powerful tools for detecting molecular homologies is the comparison of DNA sequences. Genes and their encoded proteins can be compared across different species to identify similarities and differences. For example, the genetic code, which dictates the synthesis of proteins, is nearly universal among all living organisms. This universality suggests that all life shares a common evolutionary origin. Additionally, specific genes, such as those involved in fundamental cellular processes, are highly conserved across diverse species. For instance, the genes encoding ribosomal RNA, rRNA, are remarkably similar in all organisms, underscoring their fundamental role in cellular function and common ancestry. Genetic evidence for common ancestry. Genetic evidence provides compelling support for the theory of evolution by demonstrating the relationships among species based on their genetic makeup. Phylogenetic trees, or evolutionary trees, are constructed using genetic data to illustrate the evolutionary relationships among different organisms. These trees show how species are related through common ancestors and the branching patterns of evolutionary divergence. For example, the comparison of mitochondrial DNA, mtDNA, among different species has been used to trace the evolutionary history of organisms. Mitochondrial DNA, inherited maternally, evolves relatively quickly and provides a valuable tool for studying recent evolutionary events and relationships. Phylogenetic analyses of mtDNA have revealed patterns of divergence and migration, such as the relationships among different human populations and the migration patterns of early human ancestors. Molecular clock. The concept of the molecular clock uses the rate of genetic mutations to estimate the timing of evolutionary events. By comparing the number of genetic differences between species and calibrating the rate of mutation, scientists can estimate when two species diverged from a common ancestor. This approach has been used to date major evolutionary events, such as the divergence of primates from other mammals or the emergence of specific traits in various lineages. For example, molecular clock analyses have estimated the timing of the divergence between humans and chimpanzees to be around 5 to 7 million years ago. This estimate is based on the rate of genetic mutations and the accumulation of genetic differences between the two species. Molecular clock studies help to place evolutionary events within a temporal context and provide insights into the timing of key evolutionary transitions. Genetic evidence of adaptation and evolution. Genetic evidence also reveals how organisms adapt to their environments through evolutionary processes. Gene sequencing and genomic studies have identified specific genetic changes associated with adaptive traits. For instance, the genetic basis of antibiotic resistance in bacteria illustrates how mutations and genetic variations can lead to rapid evolutionary change in response to environmental pressures. The peppered moth, Bisdun bechellaria, is a well-known example of adaptive evolution in response to environmental change. During the Industrial Revolution, the prevalence of dark-colored moths increased in polluted areas due to their camouflage against sooty backgrounds, while light-colored moths were more common in unpolluted areas. Genetic studies have identified specific mutations in the moth's pigmentation genes that confer these color variations, demonstrating how natural selection can drive adaptation. Conclusion. Molecular biology and genetics have provided profound insights into the mechanisms of evolution, revealing the genetic basis of common ancestry, evolutionary relationships, and adaptation. Molecular homologies and genetic evidence support the theory of evolution by highlighting the shared genetic heritage among species and the processes driving evolutionary change. The use of molecular clocks and genomic studies further enhances our understanding of evolutionary timelines and adaptive evolution. Through these approaches, scientists continue to uncover the intricate genetic and molecular details of the evolutionary process, deepening our appreciation of the complexity and diversity of life on Earth. Adaptation and speciation. Adaptation and speciation are central concepts in evolutionary biology that explain how species evolve and diversify over time. Adaptation refers to the process by which organisms become better suited to their environments, while speciation describes how new species arise from existing ones. Understanding these processes provides insight into the dynamic nature of evolution and the mechanisms driving the diversity of life on Earth. This section explores the concepts of adaptation and speciation, their driving forces, and their implications for evolutionary theory. Adaptation. Adaptation is the process by which organisms develop traits that enhance their ability to survive and reproduce in specific environments. These traits can be morphological, physiological, or behavioral and are shaped by natural selection, which acts on existing variations within a population. Types of adaptations. Morphological adaptations. These involve physical changes in an organism's structure. For example, the long neck of the giraffe is an adaptation for reaching leaves high in trees. The beak shapes of Darwin's finches on the Galapagos Islands are another example, with different beak shapes adapted to various food sources. Physiological adaptations. These involve changes in metabolic processes or internal functions. For instance, the ability of some desert animals to conserve water through specialized kidneys is a physiological adaptation to arid environments. Behavioral adaptations. These involve changes in behavior that enhance survival. Migration in birds, hibernation in bears, and nocturnal activity in predators are examples of behavioral adaptations that help organisms cope with environmental challenges. The role of natural selection. Natural selection drives adaptation by favoring individuals with advantageous traits. These individuals are more likely to survive and reproduce, passing their beneficial traits to the next generation. Over time, these traits become more common in the population, leading to an increased overall fitness of the species. Speciation. Speciation is the process through which new species arise from existing ones. It occurs when populations of a species become reproductively isolated from each other and diverge over time, leading to the formation of distinct species. Speciation can occur through several mechanisms. Allopatric speciation. This type of speciation occurs when populations are geographically separated, preventing gene flow between them. Over time, genetic differences accumulate due to different selection pressures and mutations in each isolated population. Eventually, these genetic differences become significant enough to prevent interbreeding even if the populations come into contact again. An example of allopatric speciation is the formation of different species of squirrels in the Grand Canyon, separated by the canyon's geographical barrier. Sympatric speciation. Sympatric speciation occurs within a single geographic area where populations diverge without physical barriers. This type of speciation often results from ecological or behavioral changes that reduce gene flow. For example, changes in feeding preferences or mating behaviors can lead to the development of new species. In cichlid fish in African lakes, sympatric speciation has led to a wide variety of species with different feeding adaptations and mating rituals. Parapatric speciation. This type occurs when populations are adjacent to each other but occupy different environmental niches. Gene flow between the populations is limited, and selective pressures vary across the contact zone. Over time, these differences can lead to speciation. An example is the grass species Anthoxanthum odoratum, which shows different evolutionary paths in different soil types. Adaptive radiation. Adaptive radiation is a form of rapid speciation that occurs when a single ancestral species rapidly diversifies into a wide variety of forms to exploit different ecological niches. This process often follows a major environmental change or the colonization of a new habitat. The Darwin's finches on the Galapagos Islands are a classic example of adaptive radiation, where a single finch ancestor evolved into multiple species with different beak shapes and feeding habits to exploit various food sources on the islands. Conclusion. Adaptation and speciation are fundamental processes in evolution that drive the diversity and complexity of life on Earth. Adaptation enables organisms to become better suited to their environments through natural selection, while speciation results in the formation of new species from existing ones through mechanisms such as allopatric, sympatric, and parapatric processes. Adaptive radiation demonstrates the rapid diversification of species in response to new opportunities. Together, these processes illustrate the dynamic nature of evolution and the continuous changes that shape the living world. Understanding adaptation and speciation provides valuable insights into the mechanisms of evolutionary change and the rich diversity of life. Evidence from comparative genomics. Comparative genomics involves analyzing and comparing the genomes of different species to uncover similarities and differences that provide insights into evolutionary relationships and processes. The advent of genome sequencing technology has revolutionized our understanding of evolution by allowing scientists to explore the genetic underpinnings of adaptation, speciation, and evolutionary history. This section delves into how comparative genomics contributes to our knowledge of evolution and highlights key findings from genomic studies. Genomic similarities and evolutionary relationships. Comparative genomics reveals striking similarities in the genetic makeup of different organisms, reflecting their evolutionary relationships. By comparing the entire genomes of different species, scientists can identify conserved genes and genetic sequences that have been preserved across evolutionary time. Conserved genes and genomic features. Certain genes and genomic regions are highly conserved among diverse species due to their essential roles in fundamental biological processes. For example, genes involved in basic cellular functions, such as those encoding ribosomal proteins, are conserved across all domains of life, including bacteria, archaea, and eukaryotes. These conserved genes provide evidence of common ancestry and highlight the shared evolutionary history of life on Earth. Comparative genomic studies. Large-scale comparative genomic studies have illuminated evolutionary relationships among species. For instance, the comparison of human and chimpanzee genomes has revealed that these species share approximately 98 to 99% of their DNA sequences. Despite this high degree of similarity, the small genetic differences contribute to significant differences in traits and behaviors. Such studies underscore the genetic basis of evolutionary divergence and highlight the role of small genetic changes in shaping species diversity. Genomic evidence of evolutionary processes. Comparative genomics also provides insights into the processes driving evolutionary change, such as adaptation and speciation. Adaptive evolution. Genomic studies have identified specific genetic changes associated with adaptation to different environments. For example, research on the genomes of populations of the stickleback fish has uncovered genetic variations linked to adaptations to freshwater versus marine environments. Differences in genes related to body armor, scale formation, and osmoregulation illustrate how genetic changes contribute to adaptation. Speciation genomics. Comparative genomics can elucidate the genetic mechanisms underlying speciation. The analysis of genomic data from closely related species or populations can reveal patterns of genetic divergence and identify regions of the genome that contribute to reproductive isolation. For example, studies of the fruit fly species Drosophila have shown that specific genomic regions involved in mating preferences and reproductive isolation contribute to the formation of new species. Genome evolution and structural variations. In addition to sequence comparisons, comparative genomics examines structural variations in genomes that contribute to evolutionary change. Structural variations include changes in chromosome number, arrangement, or large-scale deletions and duplications of genomic regions. Chromosomal changes. Comparative genomics has revealed that changes in chromosome structure and number can play a role in speciation and evolutionary divergence. For instance, the comparison of human and primate genomes has shown differences in chromosome number and arrangement, which may contribute to species-specific traits and reproductive barriers. Gene duplication and diversification. Gene duplication is a significant source of genetic innovation. Comparative genomic studies have identified instances where gene duplications lead to the evolution of new gene functions or the specialization of existing functions. For example, the duplication of olfactory receptor genes in certain mammalian lineages has been linked to enhanced olfactory capabilities. Comparative genomics in understanding human evolution. Comparative genomics has provided valuable insights into human evolution by comparing the human genome with those of other primates and even non-primate species. Studies of ancient genomes, such as those of Neanderthals and Denisovans, have shed light on human evolutionary history, migration patterns, and interactions with other hominin species. Human ancestor comparisons. Analysis of ancient and modern human genomes has revealed evidence of gene flow between humans and archaic hominins, such as Neanderthals. These genetic exchanges have influenced various traits and susceptibilities to diseases in contemporary human populations. Evolution of unique human traits. Comparative genomics has identified genetic changes associated with uniquely human traits, such as cognitive abilities and language development. By comparing human genomes with those of other primates, scientists can pinpoint genetic variants that contribute to the distinct features of human evolution. Conclusion. Comparative genomics has revolutionized our understanding of evolution by providing deep insights into genetic similarities, evolutionary relationships, and the processes driving adaptation and speciation. By analyzing and comparing genomes across species, scientists uncover the genetic basis of evolutionary change, structural variations, and the intricate history of life on Earth. These genomic studies not only enhance our knowledge of evolutionary mechanisms, but also shed light on the genetic underpinnings of species diversity and the evolutionary processes that shape the living world. Evolutionary Developmental Biology, EvoDevo. Evolutionary developmental biology, or EvoDevo, is a field that explores the relationship between evolutionary changes and developmental processes. It examines how modifications in the developmental pathways of organisms can lead to the diversity of forms observed in the natural world. By studying how developmental mechanisms evolve and contribute to the evolution of species, EvoDevo provides a deeper understanding of how complex traits and structures arise. This section explores the key concepts of EvoDevo and how they enhance our understanding of evolution. The integration of development and evolution. EvoDevo integrates the study of developmental biology with evolutionary theory, focusing on how changes in developmental processes drive evolutionary change. Traditional evolutionary biology often emphasized changes in adult phenotypes, but EvoDevo shifts the focus to how developmental processes generate these phenotypic variations. Developmental pathways. Developmental pathways are the series of genetic and cellular events that lead to the formation of an organism's body plan and structure. EvoDevo examines how these pathways can be modified through genetic changes to produce different morphological outcomes. For example, changes in the expression of developmental genes can lead to variations in body size, shape, and structure. Developmental genes and evolution. Certain genes play crucial roles in regulating development and can have significant impacts on evolutionary changes. Hox genes, for instance, are a group of regulatory genes that determine the body plan of an organism by specifying the identity and position of body segments. Changes in the expression or function of Hox genes can lead to dramatic alterations in body structure. For example, mutations in Hox genes have been implicated in the evolution of diverse body plans in arthropods and vertebrates. Key concepts in EvoDevo. Several key concepts in EvoDevo illustrate how developmental processes contribute to evolutionary change. Modularity. Modularity refers to the idea that developmental processes can be divided into distinct, semi-independent units or modules. These modules can evolve independently, allowing for the evolution of new traits or structures. For example, changes in the development of limb structures can occur independently of changes in other body parts, leading to diverse limb forms across different species. Plasticity and evolution. Developmental plasticity is the ability of an organism to produce different phenotypes in response to environmental conditions. EvoDevo explores how plasticity can influence evolutionary outcomes by allowing organisms to adapt to varying environments. For instance, certain traits may exhibit plasticity, allowing organisms to cope with environmental changes without immediate genetic changes. Over time, plastic responses can become fixed through genetic evolution. Gene regulatory networks. EvoDevo investigates how networks of genes regulate developmental processes. These networks involve interactions between different genes and their products, which collectively control the development of specific traits. Changes in the regulatory networks can lead to novel developmental outcomes and evolutionary innovations. For example, changes in the gene regulatory network controlling wing development in insects have led to the evolution of diverse wing forms and functions. Evolution of developmental processes. EvoDevo also examines how the evolution of developmental processes can lead to the emergence of new traits and species. Understanding how developmental changes contribute to evolutionary innovations can provide insights into the origins of complex structures and functions. Developmental innovations. EvoDevo studies how new developmental mechanisms and structures arise. For example, the evolution of the vertebrate jaw involved changes in developmental processes that led to the modification of gill arches into jawbones. This innovation allowed vertebrates to exploit new feeding strategies and contributed to their evolutionary success. Conservation and divergence. EvoDevo explores how conserved developmental processes can be modified to produce diverse forms. While many developmental processes are conserved across species, specific changes in gene expression or regulatory interactions can lead to divergence in morphology. For example, the conserved developmental pathways controlling eye formation can be modified to produce different types of eyes in various species, such as compound eyes in insects and camera-like eyes in vertebrates. Case studies and applications. Several case studies in EvoDevo illustrate how developmental changes contribute to evolutionary diversity. Drosophila wing development. Research on Drosophila, fruit flies, has revealed how changes in wing development genes can lead to variations in wing size and shape. These studies highlight the role of developmental genes in generating evolutionary diversity in wing morphology. Snake limbs. EvoDevo research on snakes has explored how the loss of limb development genes contributed to the evolution of limblessness. Changes in the expression of genes involved in limb development have led to the adaptation of snakes to burrowing and slithering lifestyles. Conclusion. Evolutionary developmental biology, EvoDevo, provides valuable insights into how developmental processes contribute to evolutionary change. By examining the interaction between development and evolution, EvoDevo uncovers the mechanisms driving the diversity of life and the origins of complex traits. Understanding how developmental pathways evolve and generate morphological diversity enhances our comprehension of the evolutionary process and highlights the intricate relationship between development and evolution. The future of evolutionary research. The field of evolutionary biology is continuously evolving, with new discoveries and technological advancements shaping our understanding of how life diversifies and adapts. As researchers delve deeper into the complexities of evolution, emerging technologies and interdisciplinary approaches promise to provide even more profound insights into evolutionary processes. This section explores the future directions of evolutionary research, highlighting key areas of focus and the potential impacts on our understanding of evolution. Advances in genomic technologies. High-throughput sequencing. The development of high-throughput sequencing technologies has revolutionized evolutionary research by enabling the rapid and cost-effective sequencing of entire genomes. These technologies allow scientists to compare the genomes of numerous species, providing a wealth of data on genetic variation, evolutionary relationships, and the genetic basis of adaptation. Future advancements in sequencing technologies are likely to enhance our ability to study evolutionary processes in greater detail, including the analysis of non-model organisms and ancient DNA. Epigenomics. Epigenomics is the study of epigenetic changes, modifications to DNA and histone proteins that affect gene expression without altering the underlying DNA sequence. Epigenetic changes can be influenced by environmental factors and may play a role in adaptation and evolutionary processes. Research into epigenetic mechanisms and their role in evolution is expected to provide new insights into how organisms adapt to changing environments and how these changes are inherited. Integration of evolutionary and developmental biology. EvoDevo innovations. The integration of evolutionary and developmental biology, or EvoDevo, is expected to yield new discoveries about how developmental processes influence evolutionary change. Future research may uncover additional mechanisms through which developmental pathways contribute to the evolution of complex traits and structures. Advances in imaging techniques and molecular tools will likely enhance our understanding of developmental changes and their impact on evolutionary outcomes. Synthetic biology. Synthetic biology aims to design and construct new biological parts, devices, and systems or redesign existing ones for useful purposes. This interdisciplinary field combines principles from genetics, engineering, and evolutionary biology. By synthesizing novel genetic circuits and developmental pathways, researchers can explore how artificial changes affect development and evolution, providing insights into the fundamental principles of biological design and evolution. Understanding evolutionary dynamics. Adaptive evolution. Research into adaptive evolution focuses on understanding how populations respond to environmental changes through natural selection. Future studies may use advanced modeling techniques and empirical data to explore the dynamics of adaptation in real time and across diverse environments. Investigating how organisms adapt to rapid environmental changes, such as climate change, will be crucial for understanding the limits of adaptive potential and the resilience of species. Speciation mechanisms. The mechanisms of speciation, particularly in complex and dynamic environments, will continue to be a major area of research. Advances in genomic and ecological approaches will help elucidate the processes driving speciation, including the role of gene flow, genetic divergence, and ecological interactions. Understanding how new species arise and persist will enhance our comprehension of biodiversity and the evolutionary processes that shape it. Applications of evolutionary research. Medical and agricultural applications. Evolutionary research has practical applications in medicine and agriculture. For example, studying the evolution of pathogens can inform strategies for combating antibiotic resistance and developing new treatments. In agriculture, understanding the evolutionary dynamics of crops and pests can improve breeding programs and pest management strategies. Future research may lead to the development of novel approaches for addressing challenges in health and agriculture through an evolutionary perspective. Conservation biology. Evolutionary research plays a critical role in conservation biology by informing strategies for preserving biodiversity and managing endangered species. Understanding the evolutionary history and genetic diversity of species can guide conservation efforts, enhance captive breeding programs, and inform habitat restoration initiatives. Future research may focus on developing new methods for conserving genetic diversity and promoting evolutionary resilience in changing environments. Interdisciplinary approaches. Collaborative research. The future of evolutionary research will increasingly involve interdisciplinary collaboration, combining insights from genomics, developmental biology, ecology, paleontology, and computational modeling. Integrating data and approaches from multiple fields will provide a more comprehensive understanding of evolutionary processes and address complex questions about the origins and diversification of life. Public engagement and education. Advancing public understanding of evolution and its implications for various fields will be essential for fostering appreciation and support for scientific research. Efforts to communicate evolutionary concepts and discoveries to the public will help promote science literacy and inform decision-making on issues related to health, environment, and biodiversity. Conclusion. The future of evolutionary research holds immense promise for expanding our understanding of how life evolves and adapts. Advances in genomic technologies, integration of developmental biology, and interdisciplinary approaches will drive new discoveries and insights into evolutionary processes. As researchers continue to explore the complexities of evolution, the knowledge gained will have far-reaching implications for fields ranging from medicine and agriculture to conservation and public education. Embracing these advancements and fostering interdisciplinary collaboration will be key to unraveling the mysteries of evolution and addressing the challenges of the future.